Assalamu alaikum. In this particular video, I am reviewing the complete life cycle of bacteriophage. Bacteriophages or phages are viruses that specifically infect bacteria. Understanding their life cycle is crucial for various applications in microbiology, biotechnology, and medicine, particularly in phage therapy. This document outlines the stages of the bacteriophage life cycle, detailing how these viruses replicate and propagate within bacterial hosts. There are different stages of the bacteriophage life cycle. First is the attachment. The life cycle begins when a bacteriophage encounters a susceptible bacterial host. The phage attaches to the bacterial cell surface through specific interaction between viral proteins and receptor sites on the bacterial membrane. This specificity is crucial as it determines which bacteria a particular phage can infect. Second is the penetration. Once attached, the phage injects its genetic material, DNA or RNA, into the bacterial cell. This process often involves the contraction of phage's tail, which creates a channel through the bacterial cell wall, allowing the viral genome to enter the host. Third stage is the replication. After the genetic material has been injected, the phage takes over the bacterial machinery. The host's cellular processes are redirected to replicate the phage's genetic material and produce phage proteins. This stage can vary in duration depending on the type of phage and the conditions of the host environment. Fourth is the assembly stage. Following replications, new phage particles are assembled within the bacterial cell. The newly synthesized viral genomes are packaged into capsids, and additional structure proteins are incorporated to form complete virions. This assembly process is highly efficient and can produce hundreds of new phage particles from a single infection. Fifth stage is the lysis stage. The final stage of the life cycle involves the release of new phage particles. The bacteriophage produces enzymes such as lysozymes that degrade the bacterial cell wall, leading to cell lysis or bursting. This release allows the newly formed phages to infect neighboring bacterial cells, continuing the cycle of infection. Sixth stage is the lysogenic cycle. Some bacteriophages can enter a lysogenic cycle instead of immediately lysing the host. In this cycle, the phage DNA integrates into the bacterial chromosomes, becoming a prophage. The bacterial cell can continue to divide, passing the prophage to daughter cells. Under certain conditions, the prophage can reactivate, entering the lytic cycle and leading to cell lysis. Conclusion. The life cycle of bacteriophages is a fascinating process that highlights the intricate interaction between viruses and bacterial hosts. Understanding these stages not only enhances our knowledge of viral biology but also opens avenues of innovative applications in treating bacterial infections and advancing genetic engineering techniques.